see. Okay, you can hear me? Good. Okay, you can see. Perfect. So thank you so much for that wonderful introduction. Yes, it is Women's Month, March. And thank you for inviting me, Bengt and Lori, to speak at this event. It's great to be here. And I wanted to check again. We checked in the morning, but who are high school students here? 1, 2, all of you, right? Anyone in the back? Because I will mostly speak to you. Thank you for coming. And I think this whole meeting is for you to try to inspire you to maybe pursue scientific careers. I will talk about protein folding as the basis of life and death, and you will understand that throughout my talk. And my name is Pernilla, and I'm from a university in Sweden. So when we think about living organisms, human beings, animals, everything living around us, it's built up of cells, millions and millions of cells that hold these organisms together. And in those cells and outside are proteins. And we have a lot of protein in a human being. It's about 20% of each of you is protein. So we have lots of proteins, and they are the molecular machines of life. They do all the work. And this is where my research centers. And we are going to talk now, during this 40 minutes maybe, about proteins. But I need to point out water, right? The name of the conference is The Nature of Water. And water, as you heard, is essential for the world in many different ways, and for human beings, but also for proteins. And you heard about water molecules being inside and outside of proteins already. But there's a lot of water in humans, about 70% or 60%, depending on what part of the body you look at. So if we have 20% proteins, we have 70% water, and then the rest is a few other things. So the water has many functions. And most importantly, it's a solvent for reactions, because everything that happens in a body happens in a water-based solution. So all these millions and millions of cells are like little bags of water, with a lot of things inside, but still the solvent is water. So they surround the proteins. And we heard about that. They can also go inside the protein, right? But definitely they are outside the proteins. So water is everywhere in biology. So you have to remember that everything that I will talk about, proteins, that means that there's water all around it. But before I start to talk about proteins, I want to tell you about my background, how I went from being a young kid to becoming a professor, sometimes a crazy one. So I'll show you a map. So I grew up in Sweden. I was born in the north of Sweden. I moved to Gothenburg, a city on the west coast, when I was little. And that's where I went to high school, like you are going to high school. Some of you are undergrads. I studied at this university, Chalmers. I got an undergraduate degree in chemical engineering. Then I went to do my PhD, and I got a PhD in physical chemistry. Then I worked for Professor Bengt Norden, who is the chair of this. After that, I went to America to do a postdoc. And I spent 2 years at Caltech, working in the lab of Professor Harry Gray, who will speak a little bit later after me in the afternoon. And then after that, then you can start your independent career. So then I got a position at Tulane University in New Orleans, and I became a professor in chemistry. And I was there for 5 years, until Rice University in Houston, Texas recruited me as a professor. So then I went there for another 5 years. And at that time, I had 2 little kids, kind of like that. And me and my husband decided we should go back to Sweden. The kids should grow up in Sweden. So I got a professorship in Sweden, in the north, actually in the city where I was born. So I became a professor in chemistry in Sweden. And I stayed there for 7 years until now, 7 years ago, I moved back to Chalmers, where I got my undergraduate degree and PhD. And I really moved because they started a new department and they needed to recruit more faculty. So that's where I am and my way around this. And okay, my daughters now, they are your age, thinking about you who are in high school, they are 17 and 21 today. So proteins, that's what we're going to talk about. They must fold. What do I mean? And we heard about proteins. You actually heard several protein names in the previous talks, but nobody pointed out exactly what is the protein and what does it do. So I'll give you the basics of proteins. So proteins come from the food we eat. You all had lunch now, so we actually have a lot of proteins that are degraded in the gut into the building blocks, amino acids. So you break down proteins to amino acids. These amino acids or building blocks are taken up in the body. And then they go out through the body to all the millions and millions of cells that we have. And in each cell, you build the protein that you want to have in that cell from your building blocks. And then we have, in each cell, we have about 10,000 different proteins that perform chemical, mechanical, and physical work. In principle, they make us alive. So in all of this, the proteins need to fold. And I think you've heard about the central dogma, where information in DNA is transcribed to RNA that's translated to a polypeptide chain. So you make this polypeptide chain, and I many times use the rubric snakes to illustrate this. But this chain of amino acids in a particular order, defined by the DNA, needs to fold up into a compact structure. That's what we call folding. There's no chemical bonds that are broken or formed here. It's just conformational changes. Then you make it folded, a compact structure. This is the active protein that can function. And if this protein doesn't fold the right way, it can misfold, then you can have aggregation, degradation, and that can lead to loss of function. So this type of conformational changes where proteins fold up, that's where my research area is. So I just have two actually with me of these rubric snakes here, and they can fold up into different shapes, but you have a lot of different shapes for different proteins, and it's really the shape of the protein that dictates what function it will have. So therefore, different proteins have different shapes, and they do different things. There can be problems with protein folding because there are mutations in DNA. You can see behind this that if you put in the wrong, if the DNA is changed, so you put in the wrong amino acid in a particular place in this chain, the chain might not fold the right way. Or there can be external perturbations, such as stress and aging, that can affect proteins and their shape. But normally, protein folding is spontaneous. It happens in you all the time. As I said, you had some protein at for lunch, and some of those amino acids are now made into new chains, and they are folding up as we sit here. So you go from this unfolded state, after the protein chain is released at the ribosome, to the folded state, which is the active structure. So I sometimes mimic this as, you go from a boiled spaghetti, something really floppy, you all know boiled spaghetti, right, to kind of roll up into a yarn ball. Or you can use, as I show here, with the snakes, right? And the folded state is the most stable state as physiological conditions. So that's why this reaction is spontaneous. Energetically, you want to come to the folded state. And all the information for how to fold is encoded in this linear chain of amino acids. So the question is really, how does a boiled spaghetti know what yarn ball to fold into? How does this work? All the information is in this particular chain. And we know that folding is fast. If it were a random process, it would take forever. People have calculated on this because there's so many degrees of freedom in every amino acid, every bond here, that if you just try all kinds of conformations, it will take millions and millions of years to find, by serendipity, the right structure. So there must be pathways or guiding factors. And that's what we're trying to find out. And we can find this out, or learn about it at least, by doing experiments where we look at different model systems, small proteins, simple proteins, that we look at the folding reaction. We look how fast it takes. We can look at speed, the mechanism, energetics, intermediate structures, to try to understand these reactions. Most of the time, folding works, and that's good, but sometimes there are problems with protein folding, and that can cause human disease. So that's one of the reasons why this is very important to understand more about. So problems with folding can lead to loss of function. The folded protein is not made properly. Then you don't have that function. And this is many times the case in cancer. You have tumor suppressors, and they are mutated. And when those proteins are not functioning, they're not suppressing cancer, cancer can just grow. But you also have gain of function type of diseases, or amyloid diseases, where proteins aggregate into ordered fibers, or plaque, which have a beautiful what's called cross-beta sheet structure. But here, many of these polypeptides come together and form these really, really long fibers. It's kind of stacking up. I only have two, but if you stack up a bunch of these rubric snakes, and here's the beautiful structures, all beta strands packed on top of each other. So it's not just a disordered state. It's something very specific. But you could mimic it or think about it in more real-life things. If you think about when you boil an egg, you have all the proteins in the egg white. When you heat up the egg, those proteins are unfolded, and then they start to aggregate with each other. So then when you cool down the egg, it's hard. And that's because those aggregated proteins cannot resolve and come back to that folded state. And that's why it's actually, we like that, because it's easier to eat a boiled egg than an unboiled egg. But if this starts to happen in the brain, it's not good. So these amyloid diseases are the basis of, for example, Alzheimer's, Huntington, Parkinson's, type 2 diabetes, prion diseases, ALS. You've heard about these diseases. And there are up to 50 diseases we know of now that involves amyloid formation. And this is a huge problem for the world. Age is the largest risk factor, and the population is getting older, and that's good. People live longer and longer, but we want to have a healthy life, right? We want to remember what we are doing. And for these diseases, there are no cures. There are only some treatments that can treat symptoms for some time. So this is a big, big problem that just will become, hit hard on society unless we figure things out. And the first step to reveal why things go wrong in these diseases is to at least understand how does correct folding work. So over the years, I have studied protein folding, how does it work in general, for many, many years. And others too. But what defines how proteins fold to the right structure? And of course, here water comes in. You have something called the hydrophobic effect, that most hydrophobic residues want to be on the inside and hydrophilic on the outside. But that's not all that matters, because that wouldn't result in specific structures. So during the last 20, 25, 30 years, there have been many studies of individual proteins in test tubes trying to understand these reactions. And we have learned a lot about mechanisms and speeds, how fast it can go for different smaller proteins, and what amino acids are important in the transition state and so on. And in fact, I have the world record of fastest folding protein. That was one protein that I studied when I was in Harry Gray's lab that folded in only 1 microsecond. So it was really, really fast. But if you think about the proteins that we have in our bodies, they are many times more complicated than one single polypeptide chain. So we also need to consider protein-protein interactions when you have oligomeric proteins, say a dimer. When does these polypeptide chains fold? Do they fold first and then find each other? Or do they find each other in the unfolded state, and that triggers folding? Then you have binding of metals for metal binding proteins. Here we have a little metal. It's tiny, but it's red. many proteins in our body binds to metals. Almost half of all the proteins we have interact with the small metal ion, copper, iron, zinc, for example, to add functionality. Because metals are really reactive and can be really useful. When does the metal bind? Does it bind before folding and can actually trigger folding? Or does all the proteins have to fold first and the metal comes in after? Nobody has asked those questions before. So I set out to work on this when I started my own research lab. So I have to say about the metals too that metal ions, they are a tiny fraction of that body I showed in the beginning of 70% water and 20% proteins. But we need tiny amounts of metals, but they're essential. We need the right amount, not too much, not too little. And free metal ions are most of the time toxic because they can create reactive oxygen species that can damage cells. So the cells have to be very careful how they handle metal ions. So I started out to work on these more complicated proteins to learn about their folding processes. And I want to give an example that everybody can understand so you realize how common or important this is. If you think about blood, blood is red, right? Why is it red? Because it's iron in blood. And iron is found in this protein, hemoglobin. Hemoglobin has 4 iron groups and 4 polypeptide chains in its functional state. So it's an example, it's a very common protein, rather important, that has both many polypeptides and metal ions. So over the years, we studied many different protein systems, proteins with different shapes, different metal ions, different number of polypeptides, and we figured out that protein-protein binding can precede folding for oligomeric proteins. So it's important to take that into account. And metal binding before folding can both affect the folding reaction and how fast it goes. So if we want to understand how proteins fold in real life, we need to take these complications or more complex interactions into account. Also, I just said we want to understand protein folding in real life. Then we have to think about the environment in the cell, in this bag of water. Cells are highly crowded with many biomolecules. Here's a picture where you can see a lot of proteins in the cell that are just packed together, right? They just bump into each other. This is a very, very crowded environment. And it's not just a bunch of proteins. There could be carbohydrates, there could be cell membranes, there could be lots of different things in a cell. So it's not this dilute test tube solution that we like to use in our experiments in the lab that makes it easy to see this one protein that we study. So this results in less space in a cell compared to in a test tube. And of course, this will affect water properties as well. So then we did experiments in test tubes still, but we mimicked the crowded cell environment to try to understand how is that cell environment affecting protein folding. And we found that it can affect folding speed, pathways as well, and it can also affect the final folded structure of a protein. So the question now also is, what shape does protein have in a test tube versus in the real environment? And all these results led us to ask new questions. One question is, how are metals delivered to the right proteins in a cell without toxic side effects? If the metal is important for function and folding, but how does it get there? I just said there's no free metals in a cell. So actually, we have a lot of delivery systems that move the metals. It's other proteins that help the metal go to the right protein. How does that work? And the other question that we asked is, there's so many possible interactions that can happen in these cells, right, with so many different proteins. What type of unwanted, just bumping into each other interactions happens that can cause disease? And this is very important when you think about amyloid formation and aggregation, because then a lot of proteins come together. So over the years, my research has gone from more trying to understand general principle of protein folding, correct, simple, basic protein folding, to more directly try to understand what goes wrong with proteins in human diseases. And in the lab for the last decade maybe or so, we are looking into two areas. And one is to look at the molecular mechanism for how copper ions are transported in the cell. So copper and iron are probably the two most important metals. So how is copper ions moved around in the body? And then we've learned that there's some new activities for some of these proteins here in cancer. And the other direction we do in the lab is to go from correct folding to misfolding and amyloid formation and directly look at how proteins form amyloids that result in neurodegenerative disorders and how those reactions are modulated by other biomolecules. I will tell you a bit more about the results or what type of discoveries we can do in these areas. But first I want to tell you how we study proteins. To study proteins, we need to have a protein. How do we make proteins? I mean, we can't just buy food and purify out the protein. What we do is that we make use of the central dogma, and we use bacteria as our helpers. So we put the DNA for the protein of interest into bacteria. Then the bacteria will grow, and it's just reading whatever DNA it has in its bacteria inside, in the cells, and then it makes a protein out of that. So if we grow the bacteria, after a while, you have very much of your protein. So then we have this kind of brown soup of bacterial cells. We can split them out open and then purify out our proteins. We separate things from each other with different chromatographic ways, and in the end, we get the pure protein out. So this way, we can make any protein we want, in principle. We just put the DNA into the bacteria. We can also change whatever we want of the protein. If we want to change a particular amino acid in this sequence, we can just go in and change just this one in the DNA, and then you make a new protein. And you modify the residue that maybe it binds a metal, maybe it's important for folding, and then we can test that. And then we do experiments in test tubes, mostly, which means in principle we work with bigger instruments where we put in samples and we monitor what happens with time many times. But how do we see what happens? Well, we use a lot of spectroscopic techniques, and you heard about some spectroscopic techniques already today. But in principle, the point of these type of methods is that you shine light on your sample, some kind of light, and then you look either how much of that light is absorbed by the sample or the protein in the sample, how much light is maybe emitted from the sample, or we can also twist the light to make it circularly polarized or linearly polarized, and that could give us information about the conformation of the protein in the sample. So there's many ways you can play with light to study molecules, both at equilibrium or in a time-resolved manner. We can also use biochemical techniques. Calorimetry are methods that measure heat of reaction. And more we're using microscopy. Because these amyloid fibers, this is the amyloid fibers formed in Parkinson's disease. They are big enough so you can see them under the microscope. We also do more and more cell studies. So we actually look at something living, a cell. This is a cancer cell that we try to image a particular protein in the cell. So it's kind of like doing a puzzle. You need to do a lot of experiments, and you need to put the pieces together because you see different aspects of the reaction by different techniques. So now I want to give you a glimpse of discoveries so you get a feeling for what we're looking at. And I'll talk about some copper-binding proteins, and here we'll come into breast cancer, which is the most common cancer for women. Actually, 1 of 8 will get it. So we really need to work on this. And I will talk about amyloid formation. And here I'll talk about Parkinson's disease, which is the second most common neurodegenerative disorder. Alzheimer's is the most common one. Over 10 million people in the world has Parkinson's. And in fact, every third second, think about that, a person in the world is diagnosed with a neurodegenerative disorder. Every third second. Just think about throughout my talk, how many people are diagnosed, right? So we need to better understand these amyloid processes. So let's begin with copper. So there's a protein called ATOX. It looks like that. ATOX is a cytoplasm. It's in the inside the bag of water in the cell. It moves around copper there. It's one of the key players to move copper to the right proteins. And over the years, we have studied the mechanism of this. How does this protein, ATOX, and other players in this kind of pathway of moving copper to the right protein that needs it for function and folding, how does it get there? So I thought I would show you a movie to illustrate some of the findings that we made, what we know about this pathway of normal copper transport that happens actually not just in humans, but in all organisms. So here is my movie. Oh, why does it? Okay. Good. So here you see the movie, and I'll talk because there's no sound, there's no music here. But metals come from the food, and they get into the blood. And there we have, as I said, many proteins need metals for function. And copper ions activate some really important proteins, for example, in respiration, and many other processes as well that we just need, right? So we need these copper ions. So here comes the copper ion. It goes to the cell, and you see these things that stick up there. It's a protein called CTR1 that brings the copper into the cell. And it's like a channel through the membrane. The copper comes into the inside, and then you can choose from 3 different pathways that delivers the copper to specific other proteins. But we'll follow ATOX here and care about that because that's a general pathway. It takes the copper, and then it goes to large proteins sitting in the Golgi network. So it hands over the copper, and then that other protein moves the domains so the copper comes closer to the membrane and can get through to the inside of the Golgi. So if we look at this more closely, we can see that here comes ATOX and delivers copper to these yellow spheres there, which are sulfurs. And then the metal is handed over to the next partner and the next one. So you never kind of lose the copper. Then it's on the inside in the Golgi, it goes to a protein that needs copper for folding and function. And then this protein called ceruloplasmin, it goes out of the cell because it wants to function outside the cell. And then it has its copper ions. So this ATOX is one of the key players there moving around the copper in the cell. So that's all good, right? And that's basic mechanism. But then we started to think about cancer because it's known that cancer cells need more copper ions than normal cells. And if cancer cells need more copper ions and there's no free copper, there must be copper proteins that are important. And maybe even copper transporting proteins that needs to be important. So we wanted to see if this protein ATOX was involved in cancer or in other proteins too that can bind copper. So when we looked at this protein, we could see that it was found in a cell at the front edge of a cell. And that implies that it's involved in migration. It has something to do with moving of the cell. So then we tested this. And we found, if you can look at this little movie here, these are cancer cells moving around. But if we remove the ATOX protein from the cancer cells, they can't move. So you need this protein in order to migrate or move away from your spot. And this is important because cell migration is the first step in metastasis. When the cancer is spreading to another part of your body, the cells have to move. And metastasis is really what kills cancer patients. So, I mean, this is a process that you really want to target. And then we thought, does this matter for patients? We need to think about that aspect too. So then you can go into databases And here we went in to look at breast cancer patients, and you can look at their survival. And you can look what type of proteins they had in their tumors versus survival. So if we look at this, in blue, you have patients that have little ATOX of this protein. And in red, you have patients that have a lot of the protein, ATOX, in their tumors. And you see, the red, they die much sooner than the blue. So more of this protein, you die earlier. Presumably, you get more metastasis, and that's why you die. So now we're trying to reveal the pathways involved in this. ATOX is not moving a cell by itself. There needs to be other players on this pathway. But what other proteins are involved? And can we go in there and stop something, inhibit something? Maybe this can be a new general approach to target metastasis, because all cancer cells need more copper. So all cancer cells might need this protein in order to move. So that's something we're working on. I want to say a few things about amyloid formation now, too. So amyloid formation, I'll tell you about alpha-synuclein. It's the protein that forms amyloids in Parkinson's disease. But a lot of these reactions, in all these amyloid diseases, the final amyloids look rather the same, although the bad protein is different from disease to disease. But these reactions are really complicated, very much more complicated than these simple reactions I talked about in the beginning, right? That you fold and you unfold, and you can go back and forth, and you can do all these spectroscopic experiments, and you can look at, really look at details. Amyloid formation, you go from nanometer monomers to micrometer fibers. It might be thousands of monomers in a long fiber. So these monomers have to start to assemble to intermediate species or oligomers, and they need to get longer, and it's very heterogeneous. You can have off-pathway aggregates or dead-end species, and it's a mess in principle. And also these amyloid, the long amyloids, and maybe smaller species too, can transfer from cell to cell. So that's how the disease is spreading, often in the brain. So we want to figure out what are mechanisms here. How does these proteins come together? What is starting it? What are intermediates? And of those, what is toxic? Because we really need to figure out what kind of damage do they do. And what are cross-reactivity with other species in a cell? If you assemble thousands of proteins, right, you might get some other proteins in there too, and other proteins might trigger the whole thing. So we've looked at many other things, how they can affect this amyloid formation. Other amyloid proteins, there's cross-reactivity between these different diseases. The Alzheimer's peptide can affect the Parkinson's protein and so on. Metal ions actually speed up these reactions many times. We looked at lipid membranes because that's also a big chunk of the cell that is, you can bump into a membrane. And we also tried to look at smaller molecules, design molecules that can target this and stop this at different stages. But what I want to tell you about now is external factors, not interactions with things from the outside, you could say. And there are ideas, many studies that have shown or indicated that Parkinson's might start in the gut. And in the gut, we have a lot of bacteria. In fact, we have more bacterial cells than we have human cells in a body, which is kind of amazing. So bacteria, we need the bacteria in the gut. They help us. They metabolize some things that we can't make ourselves. So some kind of symbiosis. But there are also bacteria that can make amyloid proteins. Bacteria use amyloids to stick to surfaces and so on. So they are kind of functional amyloids. But we found that some of these bacterial amyloids can affect the human proteins and both hinder or speed up their aggregation. So there's more to look into there, if we can modulate bacteria in the gut to affect these type of diseases. But we started to think about food. So there's a lot of, food has protein, and there are many food proteins that can form amyloids. Are they good for us, scary for us, or what happens to them? So then we looked into this one protein. It's called, I shouldn't do that. The protein is called parvalbumin. It's an abundant protein in fish. It's an allergen, which means that this protein comes out into the blood. So if you're allergic to fish, anybody allergic to fish? I guess a few, you could be. Then you make antibodies to this protein. So we know it can get out into the blood. So this protein is not, as I said before, degraded into amino acids in the gut, right? It survives the gut and gets out into the blood. And we found that this protein can form amyloids. When you lower the pH to mimic the gut, it forms amyloids. And they are more stable. They might survive the gut condition and therefore escape out into the blood. And that's why it becomes an allergen. So we thought this protein can meet human amyloidogenic proteins. What happens if we put them together? So what we found is that these parvalbumin amyloids can scavenge the Parkinson's protein. So kind of you have this fish protein amyloid, and then the human protein can just get attached on the surface. And that blocks amyloid formation of the human protein. So we kind of hinder Parkinson's, you could say, in the test tube. And it also affected the Alzheimer's peptide. That is also blocked from amyloid formation by this fish protein. So this is just one type of experiment you can do to see what happens. You see in the one to the left, you see really bright, long fibers. This is the Parkinson's protein. If you look at the one in the middle, you have the parvalbumin amyloids. They are thinner and not as bright. And then in the third one to the right, you have a mixture of both, but you only see the thin fibers, right? So then we know it's only parvalbumin fibers, and the synuclein, or the Parkinson's protein, has not formed amyloids. Yeah, this is just a fish that has lots of this protein, if you want to know that. Could be good. But the idea here is that we did all of this in the test tube, but maybe this is a concept to prevent or stop neurodegenerative diseases in the future, that just somehow mimic this. I mean, the best will be to just eat a lot of fish, right? But that's, we don't know how much you need, or if you can, use this type of concept to design something that would kind of scavenge the bad guys. This might also, to some degree, explain why we say fish is healthy. I mean, we always talk about fish being good for your brain and so on, and there's a lot of good fatty acids in fish that are helpful, but maybe proteins are not that bad too. So I only have a few more minutes, and I only have a few more slides, so that would be perfect. So I want to now give you a teaser of what my students are doing, I hope they are doing in the lab as I'm here speaking. Probably not now because it's evening in Sweden, but there's two things I'll just tell you about so you can kind of, get an idea. So one really cool thing that we started to look into is that these amyloid fibers, they are bad, they can spread from cell to cell, they can poke holes in membranes and so on. But it looks like they can also act as enzymes and do chemical reaction and catalyze chemical reactions. And this is something new that we haven't considered before for these amyloid fibers. And maybe this will affect disease progression. If they can catalyze reaction, they can change the metabolites in a cell, right? And that can maybe trigger more and more disease or less disease, we don't know. But maybe they could also have been, say, primitive enzymes in the prebiotic world, because it's rather easy to assemble things into amyloid fibers. The other thing we're looking into right now is a well-known cancer protein. It's called MIMO1. All these proteins have funny names. We found that this known cancer protein actually binds copper. And that's how it works. Nobody knew it would bind copper. But we found that it binds copper, and probably in a cancer cell that needs a lot of copper, you need this protein to scavenge if you get some free copper now and then, because you have so much turnaround on copper. It can pick up the copper and it shields it from doing any redox activity. So you protect the cancer cell. So this is good for the cancer cell, but not for us. So overall, all the things that you see I'm doing or working on is really about getting new molecular knowledge about proteins and what they do, right? in a way that can eventually help combat human disease. So take-home messages quickly. Proteins are essential and fold to unique structures in order to function. That's the life part. But then we can have misfolding and aggregation, and that can cause diseases. And that's the death part. And here, amyloid diseases are something that are important to address. And I also showed you that folded proteins can be good and bad depending on the context. The ATOX protein transports copper. That's good in general, but you don't want that in a cancer cell because it helps get metastasis going. And proteins do all of this in water. I want to remind you what I said in the beginning. Water is all over the place. And finally, we need to take into account the complexity of the cells. It's kind of, it's this old illustration here, but it's so many different things in the cell, so it's kind of a scary jungle. So therefore, we need to do test tube experiments too, to figure out mechanism and look at one thing at a time and then put it together so we understand the whole thing. But truly, the most important thing I want to kind of transmit to you today is that research is fun. We're discovering new things, and it's exciting to figure out what nobody knew before. And I want to say it's also very important, this goes really, really fast, but it's all about people and interactions. I don't do all of this on my own. It's all without my students, nothing would happen. So I want to show you my students. These are my students. These are PhD students and postdocs and researchers. I also had many high school students, undergraduate students that come and do shorter projects in the lab. That's really important and a way to understand what research is. This is my current research group. It's small. That's because I spent a lot of time the last 4 years running a gender initiative at my university, which is also very important. And then just some funding, which is, very important to keep going. Thank you very much for listening.